At ASEA, we are committed to advancing the science behind this great product. And there are so many things, exciting things that are happening. Tonight, we have a very special opportunity to hear from one of the key individuals behind this research. It is my great pleasure to welcome to our call tonight Dr. Gary Samuelson.
Thank you, Justin. It's a pleasure for me to be here. I'm excited to be able to report on some of the new scientific findings that we have just been made available to us through the Experimental Biology Conference in the group that is in the Human Performance Lab, North Carolina, headed by Dr. David Neiman. I am very pleased to be able to say that I've talked with Dr. Neiman, and he is indeed a scientist of a very high caliber, is interested in scientific development and interested in understanding this product and the influence and effect that it has on living organisms. When talking with Dr. Neiman, I found that he's very interested in understanding, first of all, what the components of the ASEA product are. And so in his poster that was presented at the Experimental Biology Conference, one of the first things that he mentions is that he has looked at the components of the ASEA beverage and has found that they contain these very reactive superoxides and other elements that are called redox signaling molecules, or signaling molecules in our system. This is a very significant finding because up to now there has never been a stable composition of these molecules, especially the superoxides that exist in solution. And so we indeed are now on groundbreaking science, studying what the effects of these redox signaling molecules then have on physiology, on human physiology. Another thing that he was very concerned with is the safety of the human participants in his studies. And so he went through a very comprehensive review on the safety. And we found that, he found that through him and his internal review boards, that ASEA is perfectly safe for human consumption in animals and plants and all forms of biology. In fact, there has never been an indication of toxicity, and so he is very careful when he goes about these scientific examinations to make sure that all of the participants are safe. Now, the scientific work that I'd like to be able to tell you about today, the new ones that have just come out in this past few months, are the effects of the ASEA beverage intake on the endurance performance of mice. And so in order to understand how the ASEA beverage affects our physiology, Dr. Neiman has specifically focused in on how it affects the metabolism. Our metabolism is very important, of course, to the functioning of our bodies. In our metabolism, we have two sources of fuel. One is carbohydrates and sugars, and those are digested into simple sugars that provide the immediate fuel source for our body. And part of that also is stored away in our muscle cells as glycogen, and these simple sugars then are available to our muscles immediately for the energy that we need. There's another fuel source it's very important. In fact, it's the primary fuel source of almost all of the energy we use, and that is fatty acids. These fatty acids then are stored in our body, in our fat stores, and they also come in through our foods. And they provide another source. In fact, the primary source of energy that we use, about 80% of the energy we use during the day, is through the fatty acid stores. And so this is an extremely important store of energy in our body. Now, these fatty acids then also are built up from the sugars, the excess sugars that are in our body. So if we're not immediately able to use excess sugars, they are converted over into fatty acids. And so Dr. Neiman wanted to look at this metabolic cycle to make sure that, or to see how the ASEA beverage then was affected our metabolism, and found some very interesting things. He took 60 mice in this experiment and broke them into two groups. One was an ASEA group, and over the period of 1 week, this group drank ASEA, and the other one was a placebo group that over the period of the same week drank saltwater solution. And then these mice then were implanted with some very special microtubes that are able to take fluids out of their muscles, and then placed on little treadmills, and were then caused to run to exhaustion on these treadmills. And one of the first and most interesting findings that he saw was that the mice that had been in the ASEA group were able to run 29% longer before they became exhausted. And this is a very significant finding. As he mentioned, the mice just don't run 29% longer for nothing. They have real physiological reasons why this is happening in the body. Now, in his further investigations, he looked into then several of the indicators or several of the enzymes inside the metabolism that help in the metabolism of fatty acids. And the beta-HAD enzyme was measured, and that was one that helps to convert fatty acids into energy inside this beta oxidation cycle inside our metabolism. And there's no differences seen. But phosphorylated ACC, which helps regulate how much fatty acid goes into this metabolic cycle, actually was changed. And so part of the investigation, or part of the conclusions that we might be able to derive from this, is that when ASEA is present, fatty acids and other forms of energy are more efficiently able to be converted and used inside the body. Now, if this is true, then the hypothesis is that the glycogen stores, the stores of energy, immediate energy that are available to our muscles, will be spared. That the body will use fatty acids preferentially, and glycogen will be spared. And this was another finding, or significant finding in the study, that the depletion of glycogen in the muscles of these animals in the study was 30% less than the ASEA control, than the placebo control group. So the ASEA group actually depleted their glycogen stores less rapidly. This is a very important finding, and it helps us understand. Glycogen stores, if we were to put them all together in our human body, would only provide enough energy to run maybe an hour or less than an hour, where the fatty acid stores in a typical human body would allow enough energy for us to run over 20 marathons without stopping and still have some energy left over. So the glycogen stores that are needed are spared when our body is more efficiently able to use these fatty acids. And this is a very important thing, to have more available energy to fuel the activities that we participate in. And it doesn't necessarily mean athletic activities. There's a general health benefit also related to having more available energy. our body is doing different things all day long that we don't even notice. They require energy. Our immune system is fighting off pathogens, for example. Our body is repairing and replacing damaged cells and carrying on this repair process that helps our muscles also to become repaired after athletic exertion. So all of these processes that require energy in the body can actually be beneficially affected by having more available energy to us. And I think that this is one of the largest or most important possibilities or results in the conclusions that Dr. Neiman's study makes, is that we have seen that when ASEA in an animal, in mice, is consumed, that the mice have more available energy. They can run 29% longer. They have more stores of glycogen in their muscle cells, and then the enzymes that help this beta oxidation cycle of fatty acids are also somewhat shifted, at least those which help regulate the fatty acid utilization. So we've seen some very interesting things in this study. I hope that I was able to explain it in an understandable way, and so you can see also that there are great benefits also from the available excess energy that we can obtain from drinking ASEA. Thank you.